Hey everyone, Dr. D here. Let's go ahead and get started with Chapter 8 from our Biology Concepts and Investigations textbook by Hofnagels. This chapter covers DNA replication, binary fission, and mitosis.
Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, again, welcome back. Chapter 8, we're covering DNA replication, binary fission, and mitosis. Let's go ahead and get started. Remember that all living things are made up of cells, and all cells come from pre-existing cells. So cell division is very important to understand. In fact, if you are a single cellular creature, then by dividing, that's how you reproduce, right? So imagine if you have a single cell E. coli, for instance. If it divides, it becomes 2 E. coli. That's how E. coli reproduce. It's just by dividing. And you should know, again, all cells divide. You should know that all cells come from pre-existing cells. And when we're talking about eukaryotes that are multicellular, like you and me, we have what's known as a sexual life cycle. And in this case, we grow by a process known as mitosis. However, we form what are known as the sex cells or the gametes, and those are the sperm and the egg. And the way that our bodies produce sperm and egg is through a process known as meiosis. And then the sperm fertilizes the egg to form what's known as the fertilized egg or zygote. And then through mitosis, the zygote becomes the infant. The infant grows to become the adult. Okay, so again, there's a difference in eukaryotes. We undergo mitosis in order to grow, in order to repair, in order to develop. Our cells divide by a process known as mitosis. However, in our sex organs, the testes or the ovaries, there's a process known as meiosis that occurs to form the gametes, the sperm and the egg. These are known as the sex cells, which then fertilize. The sperm fertilizes the egg to form the zygote. So we're going to learn about what happens during mitosis. We're going to learn about what happens during meiosis. And with these prokaryotes, bacteria and archaea, we're going to learn about a whole different process known as binary fission. And that's how prokaryotes divide. Okay, so we got a number of things to cover in this chapter. Again, mitosis is how our cells, eukaryotic multicellular organisms, mitosis is how we grow, it's how we develop, it's how we repair our tissues and regenerate lost body parts. Some organisms can even reproduce asexually by mitosis. Those are the single cell eukaryotes. Another process that goes hand in hand with development is called apoptosis. Apoptosis, also known as cell death, is an important process that can carve out distinctive features during development. You see, like the webbed foot of the duck. In the duck, you have a webbed foot that forms. However, in the chicken does not form the webbing, and this is due to this process of programmed cell death or apoptosis. So you should know that programmed cell death is an important part of development, and it is known as apoptosis. So that leads us to our first clicker question. That was pretty quick, right? Clicker question number 1. Shortly after fertilization, a zygote divides into 2 identical cells. What type of division is this? Is it mitotic division, meiotic division, or apoptosis? If a cell divides into 2 identical cells, that's mitotic division, okay? Remember, meiotic division is when the sperm and the eggs form. And one thing I want you to know ahead of time is that when sperm and eggs form, that's happening in the sex organs, the testes or the ovaries. and that forms the sperm and the egg. And did you know no two sperm and no two eggs have the same genetics, have the same DNA? So it can't be B. And remember, C is all about programmed cell death, so it can't be C. The answer has to be A. So let's go on to the next section here. Before cells can divide, all cells need to copy their DNA. Remember that organisms have chromosomes inside, and that makes up their genome. Their genome consists of all the cell's genetic material. Remember that bacteria usually have a single circularized chromosome of DNA, whereas eukaryotes, like you and me, we have linear chromosomes and we have numerous chromosomes. For instance, we have 46 chromosomes as humans, right? Typically. So before a cell can divide into two identical cells, we need to copy the genome, right? We need to copy all of those 46 chromosomes. We have to copy them because when the cell divides, we want to make sure that each cell has those exact 46 chromosomes. That's what genetically identical cells means. And in order to divide, when you're copying that DNA, first, the DNA replication proceeds in multiple steps. And this overview shows those steps. So let's go through these steps together. First of all, remember that DNA is a double helix with complementary base pairing between G's and C's and A's and T's. Remember that? Also, do you guys recall that DNA is directional? Do you remember that there's a 5' end on each strand of DNA has a 5' end with a phosphate group hanging off? And do you remember, can you beat Wicket? What's at the other end of the DNA? If one end is the 5' end of a strand with a phosphate group hanging off, what's at the other end? What's the other end called, and what does it have hanging off? Can you beat Wicket? That's right, Wicket is always the 3' end is the other end, and it has a hydroxyl group hanging off. Do you guys remember that? And do you remember that the two strands of DNA, you know how they say double strand DNA? The two strands of DNA are going in opposite directions. Do you remember that? That's called anti-parallel orientation, right? DNA, the two strands of DNA are anti-parallel. That means if one strand of DNA is 5' here at this end and 3' at the other strand is always going in the opposite direction. Its 3' end would be up here, and its 5' end would be down there, right? So just a recap of previously taught material. Now, let's go through these steps. Remember, I said DNA replication proceeds in multiple steps. Let's talk about these steps real quick, and then we'll go into more detail. Okay, so step 1 here, it says the strands, the double strand DNA, those two strands have to unwind. Do you see how it's unwinding? I sometimes call this unzipping the DNA, right? Unwinding the DNA. Step 2, each strand is a template, serves as a template that attracts and binds complementary nucleotides, A's, G's, C's, and T's. Remember, do you remember the complements? Again, A's pair with T's, G's pair with C's. And notice, this is on the right. Look what I'm highlighting. This is known as the template strand. This template strand serves as a template for a new daughter strand right here, you see? And on the left, look what I'm highlighting. On the left, this other template strand serves, again, as a template for a new daughter strand. So in the middle, you have these gray daughter strands, which are newly synthesized. And in the blue here, you have the template strand, the parent strand. Again, and then let's go to step 3. Each double-stranded DNA molecule consists of one original strand and one newly synthesized strand. Remember the daughter strand. As a result of what's called semi-conservative replication. This is what they call, if you ever see this term in your textbook, it's an important term to note and write down. It's called semi-conservative DNA replication. And that's exactly what it means. It means that when DNA is replicated, It's replicated in a semi-conservative fashion where you've conserved one template strand, but then you've created a new daughter strand. And then the other DNA, the other DNA molecule will also have a template strand and a newly synthesized daughter strand. That's what they mean by semi-conservative replication. Now, during this process of DNA replication, this means while DNA is being copied, there are certain enzymes and proteins that play a central role. They're listed here: helicase, single-strand binding proteins, RNA primase, DNA polymerase, and ligase. These are the 5 important proteins and enzymes that play a role. So I'm going to explain to you what each one does and what each one's role is during DNA replication. Let's go ahead and start with helicase. Step 1. Helicase separates the DNA strands. What you need to know is that helicases unwind or unzip the DNA. Next, this is step 2. Once the DNA is unwound, you see this? Look at this. When DNA is unzipped or unwound by helicase, now you have single-strand DNA. To keep the single strands apart, these single-strand binding proteins bind and prevent single strands from rejoining. So you should know that once helicase has unwound the DNA, these single-strand binding proteins attach and prevent the 2 DNA strands from reannealing or rejoining. The next player is called RNA primase or RNA polymerase. This is an important player that comes next in the process, but it's not very intuitive. There are 2 things you need to know about this step here. The first thing you need to know is that the daughter strand, right? Remember, this is the template strand in blue, and here's the other template strand in blue. And what we want to do is create a new daughter strand, right? We want to make a daughter strand here and a daughter strand here because we want to copy this template strand and we want to copy this template strand. One thing you should know is this, and you should make a note of this in your notebook, and that is that the daughter strand is always synthesized, that means made, from 5 prime. You see this? From 5 prime towards 3 prime. The daughter strand itself is made 5 prime to 3 prime, which means it's copying the template 3 prime to 5 primes. But on the exam, you should know that DNA is synthesized in a 5 prime to 3 prime direction. The daughter strand is synthesized in a 5 prime to 3 prime direction. But this is interesting, and I'll explain this later on in a little bit. Notice how the top strand is 5 prime to 3 prime towards the helicase. They call this the fork, by the way. Look at this. It looks like a little fork in the road. They call this the replication fork. And notice that this strand, the daughter strand, is being synthesized towards the fork. Whereas look at the other strand, the daughter strand is being synthesized away from the fork because it's being synthesized 5 prime to 3 prime away from the fork. And I'm going to define this later, but just as foreshadowing, the strand that's being synthesized towards the replication fork is going to be called the leading strand. And the strand that's being synthesized away from the fork is called the lagging strand, okay? This is important stuff to understand. Okay, here's the other big thing to understand. Notice that RNA polymerase, okay, primase, also known as RNA polymerase, adds a short RNA primer to template strand. So this is really weird, you guys. You would think if I'm copying this blue template DNA, I should copy it into DNA, right? I should be copying DNA into DNA. But look what Primase does. It copies DNA into RNA. This green stretch is RNA, and this green stretch is RNA. I want to take you to the board, the whiteboard, so I can explain to you just why we are adding RNA instead of DNA. It doesn't make any sense at this point, right? Why are we putting RNA down? If we're trying to copy the DNA, why are we putting a stretch of RNA with Primase? Why don't we just copy it to DNA? Well, let's go to the board where I can explain that to you.
All right, so here I'm going to explain how we copy DNA. So if we have a single strand of DNA, what you need to understand is this. Something needs to happen. You would think DNA polymerase 3 would attach to the DNA and copy the DNA, right? But that's actually not what happens. A different enzyme called, I'm going to draw it here, a different enzyme called RNA polymerase, also known as primase. This enzyme attaches first and actually copies in a 5' to 3' direction. It copies the DNA into a short stretch of RNA. Why is this? Because DNA polymerase 3 is not able to start a new strand from scratch. DNA polymerase 3 needs something to build off of. So what does RNA polymerase do? RNA polymerase attaches to the DNA and copies the DNA into RNA, a short stretch of RNA, and then the RNA polymerase goes away. All right, the RNA polymerase goes away. Next, what happens is DNA polymerase can now attach and take over because now DNA polymerase has something to build off of. You see, DNA polymerase has something to build off of. And remember, you are building 5' to 3'. You're always building the DNA 5' to 3'. Okay, so just remember when you're dealing with copying DNA, first RNA polymerase has to put down a short stretch of RNA, and this is called the primer. This short stretch of RNA is called the primer. Next, DNA polymerase 3 can take over and copy the rest of the DNA into DNA, right? Complementary DNA.
All right, welcome back from the board. I hope that was informative. So again, remember, Helicase unwinds the DNA. The single-strand binding proteins attach, preventing reannealing or rejoining of the two strands. Then Primase, or RNA polymerase, puts down a primer from 5' to 3' with respect to the daughter strand. So remember, on the leading strand, we are building towards the replication fork, and on the lagging strand, we are building away from the replication fork. And do you remember RNA polymerase, aka Primase, puts down a short RNA primer in green, and then what happens next? Do you remember from the board? Can you beat Wicket? That's right, Wicket. Next, DNA polymerase, and in the video I called it DNA polymerase 3, but in this textbook it's just called DNA polymerase, can now take over because it has something to build off of. See, the RNA primer is in green, then DNA polymerase attaches and takes over the job, building DNA. DNA is in gray. DNA is being built towards the fork on the leading strand, and on the lagging strand, again, the green is the RNA primer, and DNA polymerase 3 takes over the job and builds DNA, continuing towards the, away from the replication fork. And again, the leading strand is building towards the fork, while the lagging strand is building away from the fork. And I want to just take you to the board one more time so I can explain just what's happening during DNA replication. What is really happening during, on the leading strand on the top versus the lagging strand on the bottom. Let's go to the board where I can explain it to you a little bit more in detail.
All right, so how is DNA copied, double-stranded DNA? Let's take a look. So here we have double-stranded DNA, right? you've got your double-stranded DNA. These are the nucleotide pairings. And you want to copy the DNA. Remember, double-stranded DNA is copied in a semi-conservative fashion. So what we need to do is separate the two strands of DNA from one another. Okay, so imagine an enzyme's going to come through, cut those hydrogen bonds, separating the two strands from one another. Okay, what you're going to get is something like this. Okay, that's the 3' end of this strand, and this strand, that's the 5' end of the other strand. Remember, DNA is anti-parallel. That's why this end is the 3' end of this strand. That's the 5' end of the other strand. Okay, so we need to copy this DNA, right? So let's talk about how we're going to copy the bottom strand first. Remember, you have to synthesize DNA in a 5' to 3' direction, right? With respect to the new strand of DNA. So we need to copy which way? Are we going to copy this way, or are we going to copy this way? 5' to 3'. Actually, it would be this way, right? We'd need to go in this direction for 5' to 3' because the new daughter strand is going to be anti-parallel to this bottom strand. So, can DNA polymerase attach and start copying the DNA into complementary DNA? No. Remember what we just talked about before this? RNA polymerase. Remember, RNA polymerase must attach first. It then copies a short stretch of the DNA into RNA and then leaves, right? The RNA polymerase goes away, leaving behind a short stretch of RNA. Red is RNA. And this short stretch of RNA was built 5' to 3', and that short stretch is called the primer. The primer. Okay? But now that the RNA polymerase is gone and it's left behind the little short primer, now DNA polymerase is able to attach. Okay, let me show you that. This marker is fighting me. DNA polymerase now attaches and it copies the rest of it into DNA, right? And it just copies towards the replication fork. Okay, this bottom strand is called the leading strand, and I'll explain why in a second. It's the leading strand. What does that mean? The leading strand is the strand that is being synthesized toward the replication fork. You notice how we're going 5' to 3' towards the replication fork. The more you unzip the DNA, the more you simply copy the DNA towards the fork. So on the leading strand, you're building towards the fork in a 5' to 3' direction, and also you only need one primer. That primer's at the very beginning. Okay, so let me explain the other strand. So if I want to copy this other strand, what do I need to do? Can RNA polymerase attach here and copy towards the fork? The answer is no. If you paid attention, you know that you can only make DNA 5' to 3'. So that would be this direction, away from the fork. Does that make sense? So RNA polymerase still needs to attach, but it attaches right near the fork and it builds the RNA away from the fork. RNA polymerase then detaches and DNA polymerase can attach, and DNA polymerase now has the primer to build off of, and it will build the rest of the DNA away from the fork. Okay, that's called the lagging strand. This one's called the lagging strand. Why? Because you are lagging behind. You are moving away from the replication fork. Why? Because 5' to 3' in this case is away from the fork. All right, so what happens when you continue? Let's say I unzip even more DNA. Let's say I unzip even more of the DNA. I'm just showing you what happens when I continue to unzip the DNA. What's happening on the leading strand? That DNA polymerase doesn't care. It just keeps continuing to copy towards the fork. What happens on the lagging strand? On RNA polymerase must attach right here by the fork, build a short stretch of RNA called the primer away from the fork. DNA polymerase 3 then attaches and copies the rest into DNA. And by the way, DNA polymerase 3 will stall out here when it touches the previous primer on the previous fragment. And by the way, on the lagging strand, so every time you unzip the DNA, you got to put down a new primer and more DNA. You unzip the DNA, put down a new primer and more DNA. Okay, and you're going to leave behind these fragments of DNA and RNA. This is a fragment, and these fragments are called Okazaki fragments. Okay, all right, so in the leading strand, you put down one primer, and then you build DNA towards the fork. The more you unzip the fork, the more you copy DNA. On the lagging strand, you unzip first, then put down a primer away from the fork, and then DNA away from the fork. When you unzip more, you have to put down a whole new primer with more DNA. So you're going to end up with a bunch of these fragments of RNA DNA on the lagging strand, and those fragments are called Okazaki fragments. So I hope that helps. That's how the leading strand and the lagging strand are copied during DNA replication.
All right, welcome back from the board. I hope it made sense. Remember, so again, just as an overview, Helicase is busy unwinding the DNA. The single-strand binding proteins keep the two strands from reannealing. On the leading strand, first Primase put down an RNA primer, which is shown here in green, and then DNA polymerase constructed DNA, right, copied the template into a daughter strand of DNA from 5' to 3' towards the replication fork. On the lagging strand, remember, we had to build away from the fork. So first, Primase put down a primer, moving away from the fork. Then DNA polymerase 3 takes over and constructs the DNA. As the DNA unwinds further, we have to start a new Okazaki fragment. Do you remember? When we unwind the DNA more, a new RNA primer needs to be put down by Primase, and then DNA polymerase constructs a new stretch of DNA. And you remember this results in little fragments of primer and DNA on the lagging strand. These are known as Okazaki fragments. Now you might be wondering, what happens next? What happens after the Okazaki fragments are formed? Well, at this point, an enzyme replaces the RNA primer. Remember, the RNA primers were in green. The RNA primers are replaced with DNA, and then the enzyme ligase connects the Okazaki fragments together with covalent bonds. Ligases, what you should know is that ligases join the Okazaki fragments to one another, so you have a continuous piece of DNA. Awesome. So at this point, we understand the role of helicase, the single-strand binding proteins, RNA primase, DNA polymerase, and ligase. And we understand that DNA gets replicated in a semi-conservative fashion. There is a replication fork that's formed, and then you have a leading strand where DNA is synthesized towards the replication fork, and lagging strand where the DNA is synthesized away from the fork in these Okazaki fragments. Again, if you want to know this material in more detail, you could tune into the video above. I have a video right above my head, and that video is from my 1406. That means Biology for Science Majors lecture series from the Campbell textbook. And in that video, I go into a lot more detail about exactly how DNA replication works. So you might want to watch that video above. And as it turns out, we have so much DNA in eukaryotes that needs to be copied that DNA copying occurs at several spots at once along each chromosome. You can see this here. It says here, DNA replication has many points of origin. This is accurate for eukaryotic cells. Eukaryotic DNA has several positions along the chromosome where DNA replication occurs. Each one of these spots, see, here's one, here's one, here's one. Each one of these is known as an origin of replication. That means where DNA replication begins. And you can see here's a spot in the middle. There's a DNA fork, a replication fork proceeding to the right and one proceeding to the left. Here's some more replication forks to the left and right, left and right. And you can see here, once these DNA replication bubbles, these are called bubbles, once they touch each other, they form complete daughter strands until you've completed all of the DNA. And again, you have two identical DNA molecules, each of which is semi-conservative, remember, because it has a template dark blue strand as well as a daughter light gray strand here. And that leads us to clicker question number 2. The image at the right shows a cell's DNA before replication, so the template strands are in black. Which of the following best represents a newly created red or daughter DNA strand after replication is complete? Is it A or B? Well, again, it should be B, right? Because of semi-conservative DNA replication. The daughter DNA molecules, although they are identical, each one should have a template black strand as well as a daughter red strand. So B should be your answer there. And just touching on mastering concept question 8.2, why must DNA replicate before each cell division begins? This is because when cells divide, each cell should be genetically identical to the other, and the only way that could be possible is if we first copy all the chromosomes. We first replicate the DNA and then make sure that each daughter cell is given one set of those chromosomes. So now that we know that all cells need to copy their DNA before they divide, let's talk about the actual steps involved in cell division. We're going to start with prokaryotes. Do you guys remember prokaryotes include the bacteria and the archaea? And the way they divide, the way the cell divides is called binary fission. So it says here, in prokaryotes, reproduction occurs by binary fission, an asexual process that replicates DNA and distributes it to two daughter cells. And here you can see on this graphic on the right, the process of binary fission. So let's go through the steps involved with prokaryotic binary fission. Please recall that prokaryotes have a single circular chromosome, and you can see it here. Here's a prokaryotic cell. Let's pretend this is a bacteria. You have the nucleoid, which consists of a double-stranded circle of DNA, and it's usually a single chromosome. You have the cell membrane, the cell wall. Remember that bacteria have cell walls comprised of peptidoglycan. And here are the steps. Let's go through these steps. So first of all, as the cell prepares to divide, remember it's essential that DNA is replicated and attaches to the inside of the cell membrane. So this single circular chromosome gets replicated, remember, with all the steps we talked about before with helicase and DNA polymerase, etc. We're going to copy that DNA. That DNA sticks to the plasma membrane, and then that DNA moves to opposite ends of the cell. The cell membrane extends and grows between two DNA molecules, partitioning them off. Look, the cell membrane moves apart and it separates the two DNA molecules. And remember, these are two identical DNA molecules because this is a copy. These are copies of the same chromosome. Okay, what's the next step? Next, the cell divides in half, forming two daughter cells, each identical to the original. Why? Because the DNA was copied. So now each of these daughter cells contains one copy of that original chromosome. So these are called genetically identical daughter cells. Both have the same exact DNA sequence as one another. However, these are two different organisms now. For instance, if we had started with a single E. coli unicellular creature, now after binary fission, we have two E. coli. The E. coli have reproduced asexually. By the way, this is known as asexual reproduction because it did not require sperm or eggs or any of these gamete cells. When the daughter cells are identical, when there's reproduction and the daughters are identical, that's known as asexual reproduction. That leads us to clicker question number 3. Why is binary fission an asexual process? A, it occurs only in prokaryotes. Nope. B, it does not require DNA replication. That's not true. It does require DNA replication. C, it produces two identical cells. That's it. It has to be C. That's what asexual reproduction results in, two genetically identical cells. Now, before we go on to the next section of this chapter, let's take a quick break time with Gizmo and Wicket. I think they're in the laboratory doing some experiments at the moment. Let's check in on them for a break time and come back strong to finish off this chapter. All right, welcome back from break time with Gizmo and Wicket. Now let's continue on with how DNA replication occurs in eukaryotic cells. So unlike prokaryotic cells, which undergo binary fission, eukaryotic cells undergo a process known as mitosis. So let me explain the events leading up to and including mitosis. Now, before mitosis can take place in a eukaryotic cell, such as a skin cell on your body, before the cell can undergo mitosis and divide, it needs to prepare for cell division. And this is a process known as interphase. This is a time period known as interphase. This is when the cell is not dividing, however, the cell prepares to divide. During interphase, the cell replicates its DNA and carries out many functions unrelated to cell division. And you can see here, let's take a look at this graphic. It's showing you what you're seeing here is called the life cycle of a cell, the cell cycle. Okay, and notice how interphase, which is this giant orange arrow, interphase takes up much of the cell cycle. Interphase takes up, in fact, interphase is known to take up approximately 90% on average of the cell cycle, while the cell division itself, called mitosis, followed by cytokinesis, this is only about 10% of the cell cycle. So you could say that a eukaryotic cell spends a majority of its time in interphase, doing things unrelated to cell division, doing things like getting ready for cell division and growing. That's what's happening during interphase. And notice that interphase is broken down into 3 subphases. There's G1, S, and G2. Distinct things are occurring during these subphases of interphase. And we're going to talk about G1 and S and G2 and what they have to do with interphase and what's occurring inside of the cell during each of these subphases of interphase. During G1, the cell grows and functions normally. Your skin cell is functioning as a skin cell. Okay, G1 is a subphase of interphase. And during G1, your cell is growing. It's doing its job as a normal cell. If you block a cell from entering G1, you will enter what's known as G0. Protein synthesis occurs. the cell is doing its job. Look at this phase down here, G0 phase, where the cell is non-dividing. What's G0? G0 is actually considered exiting the cell cycle. During G0, the cell continues to function, but it is not replicating its DNA or dividing. Most of your cells are currently in G0. When the cell exits the cell cycle, it enters G0. This means that the cell is not planning on dividing anytime soon. The cell is functioning as a cell in your body, and it is not yet planning to divide. However, if the cell gets a signal that says, please divide, right? Your skin cell gets a signal to divide, that's its cue to re-enter the cell cycle. See, G0 cells re-enter the cell cycle, and they proceed past G1 into S phase of interphase. In S phase, the cell replicates its entire genome. So for instance, you and I, we have typically 46 chromosomes. Well, those 46 chromosomes get replicated. Remember the way that we just talked about a few sections ago with the helicase enzymes and DNA polymerase and replicase, right? We're going to copy all 46 chromosomes. So now we're going to have 92 chromosomes because we have 2 copies of our genome.
As you know, humans have 46 total chromosomes, right? Humans have 46 chromosomes total. But you inherited 23 of those from your dad, and you inherited 23 from your mom. All right, I'm going to use blue for dad, red for mom. Okay, the egg from your mom's egg, the egg housed 23 chromosomes. 1 chromosome 1, 1 chromosome 2, 1 chromosome 3, 1 chromosome 4. You get the idea. 23 different chromosomes. The sperm from the dad housed 23 chromosomes as well. This included a chromosome 1, a chromosome 2, a chromosome 3. So every person then has 46 total chromosomes, but they actually have 2 sets of chromosomes. What's a set? A set means 1 through 23, having chromosomes 1, chromosome 2, chromosome 3, chromosome 4. A set of chromosomes 1 through 23, that's a set. So you inherited a set from your dad, and you inherited a set from your mom, and so you have 2 sets of chromosomes. You know what that means? That means, let me draw chromosome 1, for example. Let's say this is chromosome 1 from your dad. You also have a chromosome 1 from your mom. Now, both of these are called chromosome 1. Okay? Both are chromosome 1. But are they identical? If you were to sequence the DNA, would it have the exact same sequence? No. That one's from your dad, and that one's from your mom. Your mom and dad are not related. So they're not going to have a similar DNA. It's not going to be the exact same. So these are not the exact same chromosome. They don't have the exact same sequence, but they're both called chromosome 1. Okay, why are they called chromosome 1? They're both chromosome 1, and you have 2 chromosome 2s, and you have 2 chromosome 3s, and you have 2 chromosome 4s. So what constitutes the chromosome number? Well, here's the thing. These 2 do have something in common, but it's not the exact same sequence. What they have in common is that the same types of genes that are on this chromosome are found on that chromosome. Does that make sense? You remember what chromosomes are? Chromosomes are one continuous piece of chromatin or DNA, one continuous piece. And remember, the chromosomes house the genes, right? There are genes along the chromosomes. Each chromosome contains thousands or at least hundreds, I should say, hundreds of genes on each chromosome. All right? So whatever genes appear on this chromosome 1 also appear on that chromosome 1. For example, if eye color, the gene for eye color was on this chromosome, it would also be on the other chromosome 1. Okay, if hair color was on this chromosome 1 from mom, it would also be on that chromosome 1 from your dad. So what we could say is the same types of genes appear on these chromosomes, but not the exact same sequence. Okay, so again, you have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, but they're not identical. One's from your mom, one's from your dad. But what they do share in common is that they share the same types of information, the same types of genes, okay? And there's a term for this. You see how there's the paternally inherited from your dad and the maternally inherited chromosome 1. They are called homologous chromosomes. Okay, homologous chromosomes. Homologous chromosomes are not identical. Homologous chromosomes share the same chromosome number, but remember, one was inherited from your dad, one from your mom, so they have the same types of genes on there, the same types of information, but they're not identical. Okay? Now, again, you've inherited 23 chromosomes from your dad, 23 chromosomes from your mom. You have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s. Okay? Now, what happens if the cell wanted to divide? Right? A normal cell has 46 chromosomes, right? Well, what if that cell wants to divide into 2 cells? Well, it can't just go ahead and divide, because if a cell went ahead and divided, each new daughter cell would have 23 chromosomes. Does that make sense? That's not good. You don't want your daughter cells to have 23 chromosomes each if one cell divides into 2. You want it to divide so that it retains its 46 chromosomes. So in that case, you know what you need to do? Before the cell divides, you need to copy all the DNA, don't you? You need to copy all the chromosomes. So for example, if this is dad's chromosome 1, you need to make a copy of it. You need to have 2 dad's chromosome 1s, right? See? You need to make a copy of dad's chromosome 1. So these are now identical copies of dad's chromosome 1. And then you also need to copy mom's chromosome 1. You see that? You need to make a copy of that as well. So this is an exact carbon copy of mom's chromosome 1. Now you have 2 of mom's chromosome 1, 2 of dad's chromosome 1, and you're going to have a copy of all your 46 different chromosomes. So how many total chromosomes do you have at this point? You have 92 total chromosomes, right? 92. That way, if you were to divide at this point, you would have 46. You'd be back where you started, which is what you want. Okay, there's a term for this. You need to understand this. You see how now you have a copy of every chromosome? Those copies have a very specific name. Those copies are called sister chromatids. Okay? Sister chromatids are identical in every way. Sister chromatids are identical in every way. Sister chromatids. And here's the thing. A normal skin cell doesn't have sister chromatids, right? It just has 46 chromosomes, one set from mom, one set from dad, okay? The only time a cell has sister chromatids is when the cell is about to divide. The cell has considered it is about time to divide the cell into 2. At that point, it makes a copy of all the 46 chromosomes, and only at that point do you have sister chromatids. Okay, so hopefully this was helpful. You should definitely definitely understand the difference between homologous chromosomes, which are not identical. One was inherited from mom, one was inherited from dad. They share the same chromosome number because they have the same types of genes. And then the difference between that and sister chromatids, which only exist after the DNA has been replicated. By the way, foreshadowing, this is going to happen during S phase of interphase. Okay, only at that point do you have sister chromatids. At this point, you enter S phase of interphase. This is called synthesis phase. And what you're synthesizing is DNA. At this point, a cell will take its 46 chromosomes. Remember, each human cell has 46 chromosomes inside. At this point, you will copy each and every chromosome, make a carbon copy of each and every chromosome. It's a process called DNA replication. Okay, so you're going to end up with 92 total chromosomes. I'm not drawing all the 92 chromosomes. I'm just drawing 4 chromosomes. And then during S, I'll draw another dad's chromosome 1, another dad's chromosome 2, because those got replicated. And then I'll draw another mom's chromosome 1 and another mom's chromosome 2. So now I went from 4 chromosomes total to 8 chromosomes total in my drawing, right? But just be aware that in a real cell, human cell, you would have gone from 46 total chromosomes to 92. So again, what is S phase of interphase? S phase is called synthesis. Oh, my cap. Here we go. Synthesis. Synthesis step. Okay? And what are you synthesizing? Again, you are synthesizing DNA. You copied all the DNA. Okay? In anticipation for cell division, which is coming up soon. All right? But you're not done with interphase yet. There's still one more part to interphase. It's called G2, or growth phase 2.
Now the cell is committed to dividing. At this point, the cell proceeds into what's known as G2 phase, which is another subphase of interphase. The cell produces proteins that are necessary for mitosis.
And the main thing that happens here is, you remember this, each animal cell has a centriole pair? That gets replicated. So you actually end up with 2 centriole pairs. You actually end up with 2 centriole pairs. And remember, the centrioles are made of microtubules. And actually, what's really interesting, microtubules can start being synthesized. Microtubules start growing out from these centrioles. But I'll get to that in a little bit. I'll explain it more when we get to some of the later steps of mitosis coming up. But what you should realize about G2, what you need to know about G2 is that the centrioles, oh, these caps are particularly tight today. The centrioles are replicated. Okay, the centrioles get replicated. And also, you should also be aware that the DNA, which was loose, remember the DNA is spaghettified, or chromatin. The chromosomes are comprised of chromatin, and that chromatin is DNA wrapped around those histone proteins, and that DNA was really loose, right? Like a bowl of spaghetti. Okay? At this point, that DNA can start to condense. It's not quite a mitotic chromosome yet. It hasn't fully condensed yet, but it's starting to condense. Hey! That's my cat, Tig. He's come to say hi. So anyway, I hope this makes sense. During interphase, that's 90% of the cell cycle. The cell is growing during G1. It has synthesized its DNA. You've copied all of the chromosomes in S. And then in G2, you have replicated the centrioles. And they are moving to opposite poles of the cell. The DNA is also starting to condense. By the way, one more point before the end of this video. When you've copied each and every chromosome at this point, notice that you now have 2 of mom's chromosome 1. 2 of dad's chromosome 1. you now have sister chromatids. Only after S phase do you have sister chromatids. You don't have that before, okay? Just be aware of that.
And now the cell can finally divide. The cell can exit interphase and enter mitotic phase, which includes mitosis and cytokinesis. Cells leaving G2 phase enter mitosis, the division of the nucleus, which is followed by cytokinesis, which means the division of the cell itself. And mitosis also has a series of substages as well. Mitosis is made up of prophase, prometaphase, metaphase, anaphase, and telophase. Those are the subphases or substages of mitosis. And again, mitosis, you can think of it as the division of the nucleus. So you can think of it as the division of the DNA, right? You've made 2 copies of the chromosomes. You want to line them up and you want to separate them so that each daughter cell gets a copy of each chromosome, right? That's what's happening during mitosis. And then cytokinesis is when the cell itself separates, right? The cytoplasm separates. The cell separates into 2 distinct, genetically identical daughter cells. Now, before I go into step by step what's happening during mitosis, during prophase, prometaphase, etc., I need to tell you a little bit more about how eukaryotic DNA looks and how the eukaryotic DNA transitioned from a loose state to a coiled or condensed state, because that's going to help us to understand what's happening during cell division. So take a look here. Inside of a eukaryotic cell, DNA can either be loosely packed, as you can see in this left cell. This is known as loosely packed DNA. Or look at the cell on the right. This cell on the right has condensed DNA. This is what the DNA would look like inside of a condensed DNA cell, right? So our chromosomes can either become loosely packed or condensed. So we need to understand why, and I'm going to explain that next. By the way, when DNA becomes condensed, when our 46 chromosomes become condensed into these structures that become visible, see, now we can see each of the chromosomes because they've condensed. When the chromosomes are not condensed, they are loose, and that's called Euchromatin. When the DNA is fully condensed, those are called mitotic chromosomes, or just sometimes called chromosomes. These are the structures you know and love. When you think of a chromosome, you're thinking of mitotic chromosomes. You're not thinking about Euchromatin, where the DNA is loosely packed. Now, why would the DNA be loosely packed? DNA replication and protein synthesis occur when the DNA is loosely packed. Only when the DNA is loosely packed can it be transcribed and translated. Remember gene expression, the process of making proteins from genes? That can only occur if the DNA is loosely packed. And do you guys remember when we copy a cell, when a cell divides, you have to copy the DNA and make a copy of that DNA? That can only occur when the DNA is loosely packed. However, when the cell is actually dividing, you don't want the DNA to be loosely packed. You want the DNA to be coiled up. And that's why they're called mitotic chromosomes, because the DNA coils up really tightly into distinct little bundles during mitosis, so that it's easier to move those chromosomes to the new daughter cells. Isn't that neat? So the DNA is loose when a skin cell is just being a skin cell or preparing to divide. However, the DNA becomes condensed when the skin cell is actually dividing. Again, just think of this: DNA loose during when the cell is not dividing, condensed when the cell is dividing. And what does our DNA look like, and how does it become condensed? Well, it turns out that our chromosomes are not just made of DNA, right? I think we touched on this on a previous chapter, but do you guys remember that the DNA is wrapped around these proteins called histone proteins? And that forms these DNA plus 8 histone protein structures called nucleosomes. And then the nucleosomes look like a kind of like a ball and string structure. And that makes up what's called chromatin, right? So our chromosomes consist of chromatin, which means DNA wrapped around histone proteins, right? That's what chromatin means. And again, remember the DNA condenses into these mitotic chromosomes, which look like little X's, only during cell division. When the cell is actually dividing, that's when you can see the individual condensed chromosomes inside of the cell. When the cell is not actively dividing and it's not actively in mitosis, the DNA is not condensed, and so you can't see the individual chromosomes. And it's those histone proteins that help. They help the chromosomes to condense this way. So now let's talk about what exactly is happening during each subphase of mitosis, during prophase, what's going on, and then go on from there. Again, prophase is the first subphase of mitosis. And in fact, let's go over to the board and I'll explain in detail what's happening here during prophase. Let's do that and come right back.
Notice that the cell still has a nucleus, okay? The nucleus is still there, but the DNA inside has fully condensed. They form the mitotic chromosomes, which look like these X's. And actually, if you recall, these X's are actually the two sister chromatids being held together, right? Remember at the end of interphase, you had sister chromatids, which are exact copies of the same chromosome. Well, they get held together by cohesin proteins, and they are held together as a pair of sister chromatids. So what you should realize is during prophase, the DNA condenses. The DNA condenses, and what you have are pairs of sister chromatids. So the DNA condenses into these mitotic chromosomes. You can actually visualize them at this point under a microscope. and you consider these pairs of sister chromatids. And remember, sister chromatids are identical. This is mom's chromosome 1, for example. That's also connected to mom's chromosome 1, the exact same thing. Dad's chromosome 1 is connected to dad's chromosome 1. Dad's chromosome 2 is connected to dad's chromosome 2. And mom's chromosome 2 is connected to mom's chromosome 2. So the exact same information is connected to one another with those cohesin proteins. Additionally, remember these centrioles? Remember the centrioles that were replicated previously? They're moving apart. They're moving towards opposite poles of the cell. And notice how these microtubules are growing from the centrioles. I want to introduce this concept real quick. See these little microtubules? They're growing out from the centrioles. That's called the mitotic spindle. Microtubules can be synthesized. So here in the center you have centrioles. Microtubules can grow out from those centrioles. Microtubules can radiate out, okay? And those centrioles are moving to opposite poles of the cell, and I'll explain why that's important shortly. But for now, that's what you need to know about prophase. It's the first part of mitosis, the first of 5 subphases. The DNA condenses, and what you end up with are pairs of sister chromatids. The centrioles start to move apart. Okay, and what you're forming here are microtubules that are coming out from them, and that's called the mitotic spindle.
All right, welcome back from the board again during prophase. Here's prophase as it appears in an animal cell. Here's prophase in a plant cell. Again, what's happening during prophase? The chromosomes are condensing. Those sister chromatids pair up, right? The exact copies of the chromosomes pair up. The spindle forms. Remember, this is a centriole pair. That's a centriole pair. The little protein fibers form called spindles. Spindles form. Little spindle fibers form. This is becoming what's called the centrosome. And here's another centrosome. And remember, those centrosomes move to opposite poles of the cell. So now let's see what happens during prometaphase.
The nucleus has now broken down and disappeared. It breaks down into little vesicles, okay? The nucleus is gone. So you know what that means? The sister chromatid pairs are now floating around in the cytoplasm. And not only that, notice that the centrioles, the pairs of centrioles, are now at opposite ends of the cell. They're at opposite poles of the cell. All right, and microtubules have grown. Remember the mitotic spindle, these microtubules that are growing out from the centrioles. Those microtubules have now attached to the sister chromatid pairs. Remember, sister chromatid pairs have these kinetochore proteins on the outside. And kinetochore proteins allow for docking of microtubules. Microtubules can dock onto the kinetochore protein. So these microtubules are actually attaching to the sister chromatid pairs at the narrow waist, at the centromere, at those proteins called the kinetochore. Okay? And so what do we need to know about prophase? Again, the nucleus breaks down. Nucleus breaks down. Centrioles are at opposite ends of the cell. Okay? And microtubules attach to kinetochore. To kinetochores. Okay, so microtubules are attaching to the kinetochore. The sister chromatids are now being moved. They're actually moving. The sister chromatid pairs are moving toward the center of the cell. They haven't quite made it to the center of the cell, but they are moving to the center of the cell. Okay, so again, nucleus breaks down, centrioles are at opposite ends. These spindles, these microtubules that have been synthesized from there, attach to kinetochore proteins on the sister chromatid pairs, and the sister chromatid pairs start to make their way to the center of the cell. They're not quite there yet. Let's save that for the next step.
All right, so again, prometaphase as it appears in an animal cell versus a plant cell. Remember what's happening? The nuclear envelope breaks up. The spindles from the centrosomes attach to the kinetochore on the chromosomes. So it's attaching to these sister chromatid pairs at their narrow waist. It's attaching to the kinetochore. And by the way, remember, the spindle attaches to the kinetochore proteins at the centromere, the narrow waist of the sister chromatid pair. And remember, those sisters make their way to the center of the cell, but they're not quite there yet. Next, let's talk about what happens during metaphase. The next phase is called metaphase.
During metaphase, the sister chromatid pairs have lined up down the center of the cell, down this imaginary plate called the metaphase plate. Okay, The sister chromatid pairs have now lined up down the center of the cell. And each kinetochore on each sister chromatid is attached to a microtubule. Okay, so look, again, for example, these sisters are identical copies of the same chromosome. For example, this is dad's chromosome 1. That's also a copy of dad's chromosome 1. This sister is attached to a microtubule from this end of the cell. This sister is attached to a microtubule from the other end of the cell. Okay, so the most important thing to understand is sister chromatids line up at center of a cell along the imaginary plate, the metaphase plate. Okay, so that's really all there is to metaphase. At this point, the sister chromatids have lined up, and remember that every kinetochore is attached to a microtubule from the different ends, the different centrioles. By the way, I haven't mentioned this before, but it's worth noting that at this point, they called these centrosomes. Okay, centrosome is a new vocab word to understand. That means the centrioles plus these microtubules, that would be called the centrosome. This would be called a centrosome. You no longer really refer to it at this point as centrioles because, there's more to it than just that. Okay,
And here's what metaphase looks like in an animal cell. Notice how the chromosomes are lined up down the center of the cell with centrosomes on each side of the cell. Here you can see the equivalent in a plant cell. You can see those chromosomes lined up at the center of the cell, and you can see the spindles going in either direction. So what happens during metaphase? You should know during metaphase, the sister chromatid pairs of chromosomes line up along the equator of the cell, an imaginary plate called the metaphase plate. Next is called anaphase. During anaphase, these sister chromatids, remember, these are identical copies. Here's 2 copies of dad's chromosome 1. Here's 2 copies of mom's chromosome 1. Here's 2 copies of dad's chromosome 2 and 2 copies of mom's chromosome 2. See, the copies are going to separate from one another. The sister chromatids will separate from one another, and that's coming up during anaphase next.
do you remember the sister chromatid pairs, how they had lined up down the center of the cell during metaphase? Well, do you remember that they were held together? The sisters were held together with cohesin proteins at the centromere. Well, those cohesin proteins break apart, and the sister chromatids are released from one another. Okay, look what's happened. This sister chromatid has been released from its pair, from the other sister chromatid. And those sister chromatids are moving to opposite poles of the cell. Okay, so the important takeaway to anaphase is that, this is what you need to know, sister chromatids separate from what? From one another. Sister chromatids separate from one another, right? Remember the sister chromatids were hanging out as a pair. During anaphase, the cohesin proteins holding them together let go, and the sisters move to opposite poles of the cell. That's the important thing you need to know about anaphase, okay? Anaphase.
You can really see the anaphase process here, right? Look at this. Notice that the sister chromatids have separated. One sister for each pair is headed to the cell on the left in this animal cell, and an identical copy of each chromosome is heading to the right on this animal cell. Same thing in this plant cell. Notice how the sisters have separated from one another. So the X is no longer an X, right? Those sisters have separated from one another and they are heading to opposite poles of the cell. And again, this is what it looks like in cartoon form. Notice that the sisters are heading to opposite ends of the cell. And this is why you end up with two genetically identical cells, because the exact same genetic information that's heading to one cell is also heading to the other cell. Anaphase, centromeres, that means the narrow waist of the chromosomes. The centromeres separate, right? So these two sisters separate as sister chromatids move to opposite poles of the cell. One copy of the chromosome heads to this pole, the other exact copy, identical copy, heads to the other pole. Now we're ready for telophase. Telophase, the final phase of mitosis. Let's talk about that at the board next.
By this point, remember, the sister chromatids had just separated from one another in anaphase. And in telophase, those sister chromatids have fully separated to opposite poles of the cell. So this cell has a dad's chromosome 1, and this other new daughter cell, developing daughter cell, also has an exact same copy of that dad's chromosome 1. This cell has a mom's chromosome 1. This cell has an identical mom's chromosome 1. This cell has a dad's chromosome 2. This cell also has an identical copy. You get the point. Whatever chromosomes are now in this developing daughter cell are also in this developing daughter cell. The same genetics, the same genomic information is in both developing cells. And what you should realize is during telophase, look, a new nucleus starts to form, okay? See, a nucleus starts to form around this genome, this DNA, and a new nucleus starts to form around this as well. So, what's happening? Daughter nuclei form. Two daughter nuclei form, okay? And the DNA at this point, now that the DNA has been separated, at this point the DNA is free to start to unravel, right? The DNA does not have to be so condensed. It doesn't have to exist as mitotic chromosomes. The DNA is free to relax and decondense, I should say, and become like spaghetti again. You know, remember back to chromatin structure, Euchromatin. The DNA exists as a relaxed structure. It does not exist in these tightly coiled up structures except for or outside of mitosis. The DNA is free to relax. Okay, and what you should realize is this. At the end of mitosis, what have we achieved? What is the product of mitosis? This is what you need to understand. What has happened at the end of mitosis? By the end of mitosis, you should have two genetically identical cells. Okay, it's very important that you understand this. Why? Do you remember you had 46 chromosomes originally in a human, right? You had 46 chromosomes. Then you copied each and every chromosome. Remember during interphase, you copied every single chromosome. Okay, so you had 92 chromosomes. And then those copies were the sisters in the sister chromatid pair. And what did we do? We just separated the sisters. So we sent in one exact copy to one new cell and one exact copy to the other new cell. So you should have now two cells that are genetically identical to one another and genetically identical to the parent cell that made them. Okay? So those are the important things to understand about mitosis, telophase, and I hope this makes everything clear. Okay, by the way, notice how cytokinesis has already started here. The cell has started to pinch with a cleavage furrow formation. What you should realize is this. Cytokinesis is not part of telophase, but it is concurrent. That means it can happen about the same time, okay? Before telophase is finished, oftentimes cytokinesis has begun. But cytokinesis is not part of mitosis. Cytokinesis is its own thing, but mitosis and cytokinesis together are called M phase or mitotic phase.
Now you can see during telophase, the daughter nucleus forms on the left and a daughter nucleus forms on the right. The DNA starts to decondense. Here it is in an animal cell, and here it is in a plant cell on the bottom. And here it is as cartoon form. You have your daughter nuclei forming, the DNA starting to decondense. Each daughter cell is going to have one of these bundles of centrioles. See, that's why each animal cell only has one bundle to begin with before those bundles replicate during G2 of interphase. But again, during telophase, what happens? The nuclear envelope and nucleolus form at each pole. The nucleolus being this dark spot here where the ribosomal RNA is made, the ribosomes are made. Chromosomes decondense and the spindle disappears. And remember, telophase is concurrent, but not the same thing as cytokinesis. Cytokinesis is now when the cell itself is going to divide. See how the nucleus divided? See how the chromosomes separated? That's mitosis. Cytokinesis is when the cell itself separates, right? So let's talk about that next. So now let's talk about cytokinesis in animal cells versus plant cells. They undergo cytokinesis in slightly different ways. In animal cells, they undergo cytokinesis by cleavage furrow formation. So let me show you how that works. Here is an animal cell, obviously at the end of telophase, and notice what's forming at the center of the cell itself, the contractile ring. The contractile ring forms, and then that contractile ring contracts, as its name suggests, forming this inward pinching of the cell membrane called the cleavage furrow. And the more this pinches, eventually it pinches the two cells off into two distinct genetically identical cells. And that's how cytokinesis occurs in the animal cell. And you can see that here under an electron microscope, a transmission electron microscope, you can see the cleavage furrow forming here. Whereas in plants, I want you to know that plants do not undergo cytokinesis by cleavage furrow. No, instead, they build a new cell wall in the center of the cell. This is called a cell plate. First, a cell plate forms, which is an immature cell wall, and then that matures into a proper cell wall. So you should know that plants undergo cytokinesis by cell plate formation, while animals undergo cytokinesis by cleavage furrow formation. And that leads us to clicker question number 4. In some fungi and slime molds, mitosis may occur without cytokinesis. Okay, mitosis means copying of the nuclei and the DNA. Cytokinesis means cytokinesis means separating of the cells. So what would you expect if you could copy the nuclei but not separate the cells? A, cells that don't contain nuclei. No, that doesn't make sense because we are copying the nuclei. B, cells that contain multiple nuclei. Wouldn't it be B? Yeah, because you're copying the nuclei with mitosis, but you're not actually separating those into individual cells. So you're going to end up with a cell with multiple nuclei, and that is the answer B. Awesome. So let's go ahead and move on to the final section of this chapter. All right, this last section is about cancer cells. Cancer cells are indeed abnormal cells. Compared to the regular body cells, cancer cells have these distinct features. The first one is they lose specialization. You know what that means? You think of a skin cell. It's specialized to be a protective layer. It's specialized to be a skin cell. However, once a skin cell becomes cancerous, let's say it becomes melanoma, it loses that specialization. It kind of goes into a primordial state, right? It loses that specialization. In fact, you can see a picture of cancerous cells on the left and normal cells on the right. Here, this is an actual picture with a scanning electron microscope of both types of cells. Another feature is that they become immortal. This means that cancerous cells can continue to divide and divide and divide without stopping. Normal cells are not immortal. Normal cells are not allowed to divide and divide and divide without stopping. And lastly, cancerous cells can regenerate the ends of their chromosomes. These are known as telomeres, whereas normal cells, they only extend the ends of their chromosomes to a certain extent. And what does this have to do with this chapter 8? Well, I just told you about mitosis, right? I told you about mitosis. And remember mitosis, the cells are in G1 and cells can exit G1 and enter G0. And do you remember, can you beat Wicket? When do cells re-enter the cell cycle in order to divide? What has to happen to a cell that's in G0 for that cell to divide? Can you beat Wicket? That's right, Wicket. The cells have to have a signal. There needs to be some kind of signal that tells the cell to divide. Otherwise, the cell should not divide. However, in cancer cells, what happens is due to genetic mutations, remember we discussed genetic mutations in a previous chapter, what happens is genes get mutated, and not just any genes, but genes responsible for telling the cell how often to divide or giving cells the signal to divide. When you mutate these genes that regulate cell division, suddenly the cell is not regulated anymore and the cell divides and divides and divides. And that's what leads to these cancer cells forming. It's usually due to gene mutations of important genes that regulate how often cells should divide. Because normal cells have what's known as cell cycle control. And the cell cycle is controlled tightly by what are known as checkpoints. There are several checkpoints in the cell cycle which regulate how often a cell should divide. There's a checkpoint between G1 and S of interphase called the G1 checkpoint. That's the main checkpoint of the cell. There's another checkpoint between S and G2 called the S checkpoint. There's another checkpoint at G2, and there's even a checkpoint in metaphase. So there are 4 key checkpoints, and the main one being the G1 checkpoint. All of these are important checkpoints, important parts of the cell cycle where the cell should wait and receive permission to proceed in cell division. What happens though is when DNA is damaged, when genes are mutated, this can cause these checkpoints to not function correctly. So imagine if the G1 checkpoint does not function, then these cells will just keep passing that checkpoint without stopping, and that's what cancer is. It's unregulated cell growth. The cells divide and divide and divide. And at some point, unfortunately, those cells can leave the central location. They can transfer to other parts of the cell. They can kind of move to other parts of the body. So once you have a lump of cells, once you have the cells dividing and dividing and dividing, it can form what's known as a tumor. A tumor is a lump of cells. These cells have grown into this lump due to unregulated cell division. If there's a capsule of connective tissue that forms around around this tumor, it forms what's known as a benign tumor, and this means that the cancer naturally stops growing and it becomes safe, right? It's no longer a threat to you. However, if the tumor progresses, it can form what's called a malignant tumor. In a malignant tumor, parts of the tumor split off and transfer, through the blood, through the arteries and the veins, your circulatory system, or even through the lymphatics of your body. If you take A&P, you'll learn about the lymph vessels. But what happens is during malignant cancer, cells branch off, cells leave the main tumor and they travel to other parts of the body, unfortunately. And that's what's called metastasis. This is when the cells, those cells that have lost control, those cancer cells, move to different parts of the body and start growing in multiple parts of the body. And this is the worst type of cancer. And again, do you remember what I said causes cancer? Mutations that alter the function of not just any gene, but key cell cycle control genes. Cancer may arise from overactive proto-oncogenes or from underactive tumor suppressor genes. So what are these genes? I'm going to explain this to you. Proto-oncogenes are genes that are very normal in your body. These are genes that when functioning correctly, they don't cause any kind of problems. In fact, they allow your cells to work correctly and divide correctly. However, when proto-oncogenes are disrupted, like let's say I cause a mutation that stops a proto-oncogene from working or makes a proto-oncogene work too quickly, it upregulates the proto-oncogene or it changes the proto-oncogene. If I mutate a proto-oncogene, those are the genes that I was talking about. Those are the genes that you really don't want to mutate because they can cause the cell to become a cancer, right? This is called transformation. When a cell goes from normal to cancerous, the cell's been transformed, and it's caused by mutation of these proto-oncogenes. And there's a whole nother set of genes called tumor suppressor genes, and these are important. Usually tumor suppressor genes are the checkpoint proteins. Remember the checkpoints? There's a checkpoint at G1, there's a checkpoint at S, there's a checkpoint in metaphase. There's all different checkpoints, and those checkpoint proteins are coded for by tumor suppressor genes. So these are very, very important genes, right, that make the tumor suppressor proteins. And if you mutate those or cause those to become underactive, that can also lead to cancer. So what do I need you to know for the exam? I need you to know that mutation of not just any gene, but mutation of proto-oncogenes and tumor suppressor genes, these are the genes that you really don't want to mess with. These are the genes that, when mutated, are shown to result in transformation, are shown to result in cancerous cell formation. And this is why you have to be careful when you get exposed to carcinogens, right? Have you ever heard of carcinogens? Carcinogens are chemicals that can cause mutations. And why do you not want mutations? Well, because if you happen to mutate a proto-oncogene or a tumor suppressor gene, that could lead to cancer. Now, unfortunately, mutations can also be inherited. You could inherit an already mutated proto-oncogene or tumor suppressor gene. In fact, BRCA1 is a gene that's frequently seen mutated in breast cancer. And not only can it be inherited, but it's even more common in particular ethnic groups. So for instance, in Ashkenazi Jews, you see a high incidence of mutation in BRCA1. And so unfortunately, you see a high number of breast cancers among this ethnicity. And again, in addition to inherited mutations, inherited cancer risk, you can also have environmental cancer risk. Remember, carcinogens are chemicals or physical agents that you could encounter that could cause mutation and could cause cancer. And what are those environmental factors that can result in mutation and cancer? Let's see. Eating saturated fats. So if you want to prevent cancers, they suggest eating fewer saturated fats, instead opting for more fruits and vegetables, maintaining a healthy body weight, getting regular exercise, definitely not using tobacco products, because tobacco is full of carcinogens, those chemicals I told you that mutate your DNA. Avoiding UV radiation and tanning beds. This can damage your DNA and result in cancer. Make sure that you test and you have early detection, by doing your routine doctor's visits. And also, lastly, by using condoms to avoid exposure to viruses known for causing cancer. Because you can also get cancer from viruses as well. Certain strains of viruses are known to cause cancer, such as HPV, certain strains of human papilloma virus, which is spread through sexual intercourse. And sometimes, unfortunately, removing tumors is not enough. When you remove tumors, you have to make sure you remove all of those cancerous cells. Any cancerous cells that are left over in the body might continue to replicate and form new tumors. We have medications such as chemotherapy and radiation, which can selectively kill cancer cells, which are physiologically different from normal cells. And now we even have immunotherapy and gene therapy, which are the newest approaches that promise to be even more selective and cause less health damage to the host as well, to the patient as well. And that leads to the end of chapter 8. I hope it was informative. Please leave me any questions you have in the comment box below, and I will catch you guys next time.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.